Probably time for us to get started, everyone. Thank you very much for attending. My name is Michael Fitzsimons, and I'm the User Services Manager at the GDC. This is our second workshop in a series where we're trying to introduce various aspects of the GDC to the community. The topic today is querying and downloading data using the GDC the GDC data portal and the GDC data transfer tool. Just before we get started, just some ground rules. Sharon already maybe mentioned them, is that you'll be muted by default during this workshop, just because there'll be a lot of people attending and we want to preserve the audio. However, my portion of the workshop, the prepared portion, is probably only going to last, less than 30 minutes for sure. So there'll be plenty of time for questions, and at that point, you would give the questions via the chat screen. So we'll have lots of time for that, and I'll answer as many as time allows. Okay, so let's get started. All right, so what are we going to talk about today? Well, as I mentioned kind of broadly, we are going to focus on getting data out of the GDC, basically. I'll kind of introduce a general scenario for why you might be trying to access data from the GDC. Then we are going to spend quite a bit of time looking at the GDC data portal and how to use the filters within it, as well as the advanced search. And then we'll spend time on actually downloading the files, as well as I'll introduce kind of how the GDC controls protected data and how you would gain access to the data in the GDC. Okay, so here's kind of a general scenario. A lot of users are interested in some variant on this. This is, maybe a particular user is interested in a couple of specific projects within the TCGA because they, are familiar with, the rich data that was present in TCGA and want to compare their own data to this. So this is a figure taken from this particular TCGA paper on the TCGA COAD and READ project. So perhaps a user is interested in downloading some of the files stored for these different projects that were used for this analysis. Maybe in order to do this comparison, which is quite an advanced one, they would need to download BAM files, which are the raw sequencing data that has been aligned, BCF, which contains the mutation calls, maps, which are another derived data format that contains mutation calls based or at the project level. Perhaps they want the RNA-Seq data, and then maybe they're also interested in clinical data. So I'll spend some time today talking about kind of what kind of clinical data you can get in the GDC. So this is kind of the motivation for this workshop, but there's lots of different variations on this for sure. Let me start at the main GDC website, just to orient you on how you get to the data portal. So this is at gdc.cancer.gov. So this is the main, the main website for the GDC, and it contains lots of interesting information and lots of high-level information on a bunch of different topics: how to access data, how to submit data, a lot of area that the support team has created for helping users. You could get to the data portal through this big button, Launch Data Portal. You can also access all of our apps, including the data portal, through this area. Before we get in the data portal, I'll just quickly also highlight this documentation area. So this has lots of really in-depth information about the GDC and the different apps, including the data portal and the data transfer tool, which we're going to focus on today. But if you want any more information on these topics or other topics about the GDC, this is a great resource. So now that you know how to get to the data portal, let's start there. so here you have the main screen for the GDC data portal. This has just really some summary information about the different types of projects, or the projects and the identity of the projects, the number of cases, the number of files that are in the GDC. This is just to give you an idea of what's here. I'll also take a moment here to also mention what kind of data you find in the GDC, since maybe not everybody on the call would be familiar with this. The GDC is not just a data repository. We actually we use the word harmonize the data. So we don't store and distribute the raw data. What we have done is realigned it. All of this is done to HT38. We use our own variant calling pipelines and our own analysis pipelines. So that's the type of files you will find here. However, just to mention, there is an area of the GDC called the Legacy Archive. This is laid out really similar to the GDC data portal, but it does contain basically the files, the exact files that were present in CG Hub for target, TCGA, CCLE. So if you ever needed those original files, they are all still hosted by the GDC, but in this different area. Okay. So that gives you a little idea of what the content here is. We're going to spend most of the time today then in this data tab, which is where you would access the data. So on the left, you have a bunch of different ways to filter the data. And on the right, you have a summary, as well as the number of cases and the number of files that these current filters apply to. So the summary tab is just that. It gives summaries of all the different projects, some of the details about the files that are here. You can look at these in tab delimited format if you want. Let's take a few minutes and kind of familiarize you with the information that we have on cases, which is the GDC word for patient. And then we'll talk a little bit about the files, and then we will discuss how to filter them. So on the case page, right now we have all of the cases that are in the GDC, but I just want to jump into one randomly to give you a sense of what kind of information we have about patients. This is a good opportunity to introduce the topic of a UUID. So all of the entities in GDC have a unique ID that applies to them. This applies to cases, any biospecimen entity, as well as files. We have the submitter ID, which is kind of the human readable version. There's also some information on what type of experimental strategies apply to this case, the data categories. Down here, we display all of the clinical and biospecimen data associated with this case. So the clinical section here highlights the clinical data that the GDC has indexed. So what does it mean for the GDC to index that data? This is a subset of the entirety of the data that was deposited in the GDC. We haven't indexed every single clinical field that was present in that original XML file that was submitted. What we have done is try to highlight particular clinical fields that we think are broadly useful across projects, since we do have lots of different projects, or increasingly more projects, and so what we've done is try to find those clinical fields that are, going to kind of span projects and be the most powerful for filtering the data. So you can kind of click through here and see some of the information that is available. This is really going to vary across projects as well. If you do want the original clinical data, that will be found here under a particular file. So this is where the raw XML file that contains every piece of clinical data that was submitted for TCGA or other projects. So nothing's lost. These are going to be the fields that I'll show you in a moment how to filter by, however. At the bottom here, we have all the biospecimen data. So as most of you are probably aware, TCGA and other projects are very particular about how they categorize these different samples and subsamples kind of within the data, and so all of that information is enumerated here. Okay, so that gives you kind of a sense of the information about cases that are here, should you want to filter by any of that information. And files, I'll just jump into a random file here as well and give you the same bit of tour of what type of information is available. Again, you're going to see this UUID that is available for all files and data entities. You're going to see a little bit of more information how we've classified the data in this case, and I'll show you kind of those other data categories and data types that are available. You're going to see the biospecimen entity that this is associated with. You're also going to see what workflow type created this file. So as I mentioned, the GDC doesn't just store the data, it actually analyzes the data. So this particular file was created from this workflow. If we had done any further processing of this data, you would see downstream analysis files here as well. Okay, so now that I've kind of familiarized you with these tabs, let's actually apply filters, since it's very unlikely you actually want to see the entire content of the GDC. It's much more likely that you want to apply some type of filters. All right, so we have two kind of tabs over here for applying different types of filters, filters that apply to cases or other biospecimen or clinical categories, and then filters that you can apply to the files themselves. So I think we had talked about how we wanted to filter for two particular projects that you might be interested in. I just or filtered by TCGA. We were also going to focus on COAD and READ. So you can notice that on the right that this number of cases and files is already going down based on what we're selecting over here. At the bottom of this section, you're going to see different ways of filtering by the clinical data that's available. So you have the ability to filter by all of that clinical data that I showed you that was indexed. Some of those options are visible here, not all of them. So if you wanted to add another one that's fairly commonly asked is sample type. So you can go into this Add a Custom Case Biospecimen filter screen. So sample type is where, whether it's a tumor or a normal sample is located. And as you can see here, as you start to type, the interface will populate what it thinks you're trying to enter. This can be useful if you're not completely familiar with the GDC terminology. So we've added this other custom filter. As you can see here, you can now select, I only want files that are associated for tumors, for example, or I only want files that are associated with normal. This is, a powerful way to filter if you're interested in the raw alignment files, for example. Okay, so what kind of file filters might we be interested in? So there's a bunch of different ways we've broken down files. We've broken it down by category, type. Experimental strategy is one that's very intuitive and is often used. Let's focus on just the whole exome data right now, since we can start with that type of data. Then perhaps we're interested in seeing the BCF. And finally, let's even go more detailed. As I said, we do our own variant calling at the GDC. We actually have 4 different variant callers we use that are all listed here. I think we'll just start with VARScan2, the annotated version of VARScan2. All right, we're getting this down to something kind of a manageable number of files, but maybe we'll add a few more clinical filters. Perhaps we're interested in filtering by race. Okay, the file number is getting pretty small now. I'd like to also introduce this area of the advanced search here before we get on to actually downloading the files. So this is, as you might notice, has been kind of filling out as we do additional clicks over here. But you can also just enter directly into this area. So much like that other filtering I was showing you earlier, this also gives you suggestions. So perhaps we also want to apply a gender filter. Okay, so now the number is quite manageable, and maybe at this point you'd be interested in downloading some of these files. How would you do that? I think this is a good opportunity to talk about how one gains access to controlled access data on the GDC. So as you can see, all of these files, all these VCF files are controlled access. There's lots of files that are not controlled access, and in fact, while I am actually already logged in here, everything I've done so far, anyone in the public can go in and have done. The metadata that you've seen is not controlled access, so all of this looking around, finding the files, looking at the clinical metadata, none of that is controlled. And in fact, you can download lots of files that are not controlled access. It's typically pretty much just the VCF, some of the math files, the raw alignment files that are considered controlled. But let's talk about how you get access if you want it. I'll just kind of go back to the main screen here and show you some of the information that's available that we have on obtaining access to control data. So there's lots of information in here. I'll just use this flowchart as kind of an example for how you would gain access to data. The GDC does not have its own credentials. So we do not grant a GDC login, for example. We utilize existing logins that are already available for the NIH. So in order to gain access, the first thing you would need to do if you haven't done it already is to obtain an ERA Commons account. Once you have done that, you would then want to associate that login with the dbGaP access. So this is another organization that grants access based on projects. So you would apply specifically for TCGA access, or for target access, or for one of the additional projects that we'll have coming online in the future. Once you're granted access, this is just stating that you then can, disseminate that to other individuals in your lab, perhaps. And it's at that point that you have this setup that you're able to log into the GDC data portal, as well as use any of our applications in order to download the data. There's much more detail that you can find in some of these links here, so this area is a good resource for if you're interested in gaining access to controlled access data. Okay, so once you've gained access, you would have logged in. Now you want to download files. There's a couple different ways to do that. You could simply download an individual file. That is, if you're interested in a very small amount of data, this is a solution. It's not scalable if you want to download, hundreds or thousands of files. Let me show you a better way to do that. What I would recommend is adding these files to your cart. A very Amazon-inspired interface here, where you add files to your cart, and then you will go to checkout. Here again, you can see all the files. What you could do at this point then is download a manifest and use our data transfer tool. So this allows you to download lots of files en masse. And this is a good opportunity then to transition to talking about the data transfer tool. So I have a few slides about that, and demonstrate how that works. So I just wanted to kind of introduce the tool generally. This is a command line tool used to transfer large data sets to and from the GDC. You know, we're talking pretty much exclusively about downloading data today. The data transfer tool could also be used to upload data. If you wanted more information on that, there's more information on the website. Provided projects meet, you know, minimal criteria, data can be uploaded to the GDC for a specific project. The data transfer tool is based upon the GDC API. Much like all of the GDC applications, I didn't spend really any time talking about it, but the portal, the data transfer tool, the submission portal, all of that utilizes the GDC API. I think we'll have another workshop just dedicated to that later, but this is basically the programmatic access to all of the data. That's very well developed and very powerful way of getting to the data. The data transfer tool is supported on macOS, Ubuntu Linux, and Microsoft Windows. And if you did want to download that data, you would, or excuse me, download those, the tool itself, that could be done here. So this would be the binaries that you would download in order to install the data transfer tool. Kind of what are some of the features or benefits? This is really aimed at downloading large files and large numbers of files. So even that 30 files that I suggested, you could probably fairly easily download all of those in the cart. But once you get hundreds of files or even a few very large files, the data transfer tool is much superior than going through the browser. It provides a convenient way to download data from the GDC. One of the big benefits is that it is scriptable. So if you have an analysis pipeline that is going to pull from the GDC, the data transfer tool is a potential solution for that, in addition to using the API directly. I've already mentioned that it's in the command line. One of the main features of the data transfer tool is this ability to resume halted or incomplete downloads. So if you're downloading to your laptop perhaps a few files or a few large files and your Wi-Fi comes out, or there's some sort of network interruption, which is inevitable for very large downloads, this will notice that you've already downloaded certain files in the manifest, and you just execute the same command, and it will skip over the ones you've already downloaded. It also allows a little bit of, in the way of resource tuning, and what I mean by that is you can select the number of processes that are running, which can be one way to optimize the rate of download. Okay, so let's actually look at the data transfer tool a little bit. So once you've installed it, it functions as, so I'm in the terminal here on a Mac. This is going to be pretty similar if you had a Windows or a PC for doing this. This is the help screen that gives you some information on what different flags are available. I'll just point out a few here that are probably particularly important. One is this token flag. So in the portal, you're already logged in, but when you use this tool, you need to have a token that you can download from the portal to pass to the token, or else it will give you an error. Another really helpful one, potentially, if people, as I said about the resource tuning, some people will go in and modify this. The default is pretty good, but if you want to further optimize, sometimes increasing the number of processes can be helpful. Another really important flag is this manifest flag. So this is what you would use when I was showing you here from the cart. You would download this manifest and then supply that directly to the data transfer tool. Okay, so let's download a file so you can see how that is done. You have a couple options. As I said, you can just download a single file directly. So here you will use that universal ID that I mentioned. And you can see that if you do not supply a token for controlled access data, you will get an error. So I'm going to supply my token this time that I downloaded. Kind of went by kind of fast. But here's the GDC client download, single UUID, and then -T for the token. Then the other option is to use the manifest that I just downloaded. So go by fast again as well. But you can see here, GDC client download -M manifest -T token. And it's going to just go down the list and download these files until they're complete. Okay, so that's actually the entirety of the content that I had prepared. Finished about 25 minutes, as I said. I'm going to turn it over to questions in just a moment, but I wanted to highlight now, and I probably will again at the end, that if you want to ask questions offline, or we just get a chance to cover something that you wanted to learn more about, I encourage you to email the GDC Help Desk at this email address. The team is happy to answer any questions you have. We'll just wait a few minutes while people consider any questions they may have.
Hi, Michael. There's a question, is where is the recent XSD file for the clinical data?
The XSD, just so everybody knows, there's the XML, which is the file where the clinical data is. The XSD is kind of the format for the clinical data. Let me show you how you would get access to either of those. So, as I said, let's jump into a particular case, and I can show you where that information is. So here were the files that I had ready to download, but you can also kind of go into looking at these cases that were associated with those files. So what you want is a clinical supplement file. And you could also go directly into the file section and select that as a data type. That's going to be a slightly different format for target and TCGA. We'll select TCGA. So these are the raw XMLs that all of the original data came in through. The XSD, if that's really what you're interested in, at the top in the header of the XML is going to say what the XSD is and where you can get it. I think that's actually a GitHub link for most of them.
Okay.
Okay. All right, I see a couple questions now. We got a question about so I see that the GDC has files processed against GRCH38. Yes, are the files processed against HG19 available through the archive? And the answer is yes. So as I kind of briefly talked a little bit about at the beginning, what you see here in the data portal has all been aligned to the most recent build of the genome, the GRCH or HG38. All of the previous aligned data for TCGA or other projects is going to be in the legacy archive. So virtually all of this is HG19. I think maybe a little bit of it is maybe even HG18. But that will all be in here. So you would search for the BAM files that you would want in the legacy archive to get that data. Okay, so let me, can you show us an example of how to download an RNA-Seq data set? Sure, that is a process to gene level calls for a given TCGA project. Yep. So I think I mean, let's go to those two projects that we were interested in before. So I think we're doing COAD, READ. And the question is really about derived RNA-Seq data. Maybe I'll spend just a moment showing you where you will get the details about our pipelines in case those questions arise. So that would be back at our documentation site. And we have a lot of information about how we've done all of the processing. So specifically for RNA-Seq, you can find this information here. So this talks about, for this pipeline and others, exactly how that was done, all the tools that were used, you know, really detailed information about the flags for each of the tools. But just briefly, what we have done for RNA-Seq, pardon me, we'll select that experimental strategy. What you have there then is BAM files and also these more derived files, which are the count files, these text files. So what this is going to be for the text files, what we've done is you have the raw HT-Seq output, so this is the raw count for each gene, and then we have these normalized counts. We have SPKM and SPKM-UQ, which is even further normalized. So what you would do if you're interested in these derived data sets is select one of these, you know, apply any other filters you would have, and then download these files from your cart. All right, got lots of questions. So that's RNA-Seq. There's a question about the API. Basically, can you query using the API rather than just download? Absolutely. You can get, so all of this information, you know, is very useful if you're clicking around on individual files, but the API can be extremely powerful if you want to see lots of, you know, very specific information across all of the cases or all of the files in the GDC. You know, we do work to make the user interface helpful for spanning cases and projects and files as well, but sometimes the API is great if you have, you know, you really want it in the format that you specify, only these fields. That's the best way to do it. The way you would do something like that, and since it's really a topic for another workshop, but let me just briefly introduce the idea. The API user guide has lots and lots of information about how to apply filters, what kind of filters can be applied. So I would recommend that the user or the individual who asked this question take a look in here, but then feel free to get back to the support team if you have further questions on that. I think the follow-up to that is, can you get multiple cases into one file? For the API, you can absolutely do that. There's a couple other options if you don't want to use the API for doing that. What I have recommended to users in the past, so if you have all of this case information, for example, and you wanted information on all of these cases, there's a couple ways you could do that. So if we do go to the cart, this is going to download the metadata for these files. However, it's going to be in a JSON format, which I understand is not what most biological researchers prefer. So if you do want the original biotab files, you can get those in the legacy archive. And I can just mention that we are working to provide kind of GDC biotab files, which should be available hopefully fairly soon in this page so that you can get access to that kind of tabular formatted data. Can you open Can the open access data be downloaded without a login? Absolutely. So let me just log out. And we'll just find some data that's open access. So you can select, if you really only want, oh, sorry, it's on the Files tab. There, you can select by access level even. So this is some of that actually high-level RNA-Seq data that was asked about. So this can all be downloaded without a token. It's really just the data that we've determined can be tied back to an individual. So that's the alignment files, so the BAM files, as well as the SNP calls and some of the MAP files. It might not be obvious to everybody, but I mentioned the BAM files, which are the aligned reads. However, you can always regenerate the original FASTQ files if you want. All of the quality scores are maintained. I know that's a very common question, so I just wanted to make sure that was clear. Let's see if there was any other questions. Okay, I think I've covered all of the questions that have been asked so far. Let's just wait another couple of minutes to see if anybody has thought of any additional questions they might be interested in. While I'm waiting for that, maybe I'll just mention that in the future, in terms of things that are coming for the GDC, really I've shown you kind of a lot of work on downloading data from the GDC. In the future, we do hope to add additional resources for visualizing the data. So I hope, in the fairly near future, we could have a workshop on some of those new tools as they come online. Okay, we got another question here. I think it's another question about using the API for scripting. Script using API. Probably we need a little bit more direct interaction with the user about how to help in terms of using the GDC API in a script. Just suffice it to say that it's definitely possible to download, all of that index metadata with the API. You don't have to download any of the files in order to do that. I think that's kind of what you're getting at, but please feel free to email the support email, which I'll show again in a moment, with more follow-up questions if you have more interest in that topic. So there's another question about, I downloaded the manifest, all publicly available data for a particular primary site. Now I would like to sort them by data type. How do I do this best? So that sort of, after it's already downloaded, I think the solution I would recommend is an API call might be the best. I'm happy to, or the team is happy to help you make that, so you don't have to, necessarily learn how to use the API. I think once you've gotten the data, that might be the best solution in order to tie those file names back to a data type. I think it might be kind of laborious to go back in and look at all of those individually. So yeah, after it's been downloaded, I think that's probably the best solution. So yeah, just please put a support ticket and we're happy to help you do that. This is a good question. I am using CentOS. Is there a CentOS client? We get a lot of questions like that, and the answer is yes, there is. It is not a fully supported version, although we do have some instructions that we have found will work for installing the GDC client on CentOS. So it isn't on the main website just because it's really not considered to be a fully supported version, but we do have, our team is experienced with helping installs on CentOS. So just put in a support ticket and we're happy to help you do that, since we do know it is a community need and interest, but it isn't one of our primary supported platforms. Okay, let me just wait another moment or two here to see if there are any other questions.
There was a question about the presentation, making it available, and we are going to make the actual video available on our YouTube, and we'll send out the information on that once it is available.
Exactly, yeah. Fairly soon we should make this available on YouTube, as well as directly from the GDC website. There's a question about getting the XSDs. So it may be that in some of the older XMLs, the link is no longer accurate. So the current link is in GitHub. I think that that's accurate, that the older links are not supported. So if you do want, oh, actually it's public, so sorry, that you can look at the question from Mark Jensen there that has the updated link if you're interested. Okay, well, let me just splash the support email one more time before I go. Please, if you have additional questions, feel free to contact the support team at the GDC. We're happy to help in any way we can. Thanks so much for attending, everybody. I hope to see you all again at another workshop in the future.